Hi guys, I'm Joey and today we're going to be learning about a battle that many people are facing right now. Over 14 million people are fighting this battle every day, and the enemy is what we call cancer.
Every cell goes through life just like we do. These newborn cells are called undifferentiated cells. All cells age and grow just like we do. And as they get older, they find their purpose, and that's when they become differentiated. May it be a brain cell or a professor, with life comes death. Most cells undergo apoptosis, which is a programmed cell death. Now let's have a look at the difference between healthy and cancer cells.
Cells grow by dividing. One cell becomes two cells, and two cells become four, and so on. Healthy cells divide in a controlled manner and only when they need to, so we can grow and replace old and damaged cells. People don't really divide like cells do, unless...
Cancer cells, on the other hand, divide uncontrollably. This can be caused by point mutations in the gene. A couple of examples are insertion and deletion. Luckily, our immune system can recognize the cancer cells as abnormal
Cancer!
and attack them. This may kill some of them, but not get rid of all of them. Some of these cells can travel around the body. We can think of them as people who travel around the world and consider our body as if it was the world. Some cells can travel short distances, say from the liver to the skin, or it can also go from long distances, liver to the lungs. From a traveller's perspective, it could be a person from France going to Scotland. However, most of these cells are not permitted to go to places where they actually go. And if our immune system is working up to standards, they can be detected and be put under arrest. There are two main ways that these cells migrate. One is single cell migration and the other is collective cell migration. We can think of these as people who travel on their own or with others. There are two types of single cell migration. One is amoeboid. It is well known for its round shape and amoeboid-like movement. The other is mesenchymal. It is known for its spindle shape and its leading and lagging body. As for the collective cell migration, there are several types, but they're well known for their cell adhesion. One example is the collective chain migration, and another are the collective sheet cells. These cell adhesions keep them all intact and together. And the final example are the trail cells.
Cells that migrate individually utilize the gaps in the extracellular matrix to penetrate the tissue membrane. Single amoeboid cell migration is known to migrate independently of ECM breakdown in 3D substrate. Mesenchymal migration, on the other hand, have adhesive interactions with the ECM and requires the degradation of ECM so they can keep traveling.
As for collective cell migration, most commonly observed in epithelial cancers, due to their size, they need to degrade the extracellular matrix.
Despite the friendly faces, these cells can cause great distress for the host. They can create lumps, bumps, and yellowing of the skin.
Scientists all over the world have tried to deal with different cancers through surgical and drug interventions. After some testing and clinical trials, majority of the cancers have gone.
However, reviewing different drugs years after the first clinical uses, scientists have found drug resistance in cancer cells. Cancers have managed to escape the primary intervention, such as surgery or drug treatment, and grow at sites different from where they originate. This is known as tumor recurrence.
Oh no!
Researchers have found a number of proteins that activate the release of enzymes from tumor cells that break down the extracellular matrix. These same enzymes were found to release growth factors that promote cancer cell growth. This causes cancer cells to divide uncontrollably and release more growth factors for itself. In addition to this, other enzymes have been recognized to change the cancer cell behavior and cause transitions like epithelial mesenchymal transition, also known as EMT.
New research ideas are being proposed in combining drugs as studies show reduced resistance in combined therapy. By targeting the proteins that activate the enzymes that break down the ECM, growth factors are expected to be inhibited as well. With the addition of drugs that target DNA activators, DNA replication is inhibited and cell division does not occur.
We have high hopes for these new drug combinations to work better than the current treatment. But that's it for now. I hope you learned and enjoyed watching this video.